hear me clearly?
Yes.
Okay, I will start this webinar. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. We are delighted to welcome you for an international webinar that organized by the Food Science Student Forum or Formasip IPB University in collaboration with the Department of Food Science and Technology IPB University by the theme Polishing of a Diamond in the Rough, Microbial Bioconversion to Create Bioactive Unnatural Products. My name is Nurafi Rasna Suhaima. It is an awesome and precious chance for me to be the master of ceremony of this event on Saturday, 13 March 2021. And now, please let me welcome our speaker and participation. Welcome to our main speaker, Honorable Prof. Hiroshi Kansaki, Laboratory of Chemistry of Bioactive Compounds, Okayama University, Japan. Good afternoon or good evening, Professor. And also welcome to the head of Food Science Study Program, Department of Food Science and Technology, IPB University, Honorable Professor Dr. Harsi Dewantari Kusumaningrum. Good afternoon, Professor Harsi.
Good afternoon.
Good afternoon also to the Honorable Olaf Lecturer of Department Food Science and Technology, IPB University. And the last but not least, welcome to all participants of this event, our highest appreciation and thank you very much for your participation. So ladies and gentlemen, here is the agenda for today's webinar. First is opening session, and next welcoming remarks, and also main event that contains presentation and question and answer session. Also, there is a door prize and group photography, and the end is closing session. So first of all, before we start our agenda today, let us pray together to God according to our beliefs. Pray begins. Finished? Okay. Well, ladies and gentlemen, we need to inform you that the link of attendance will be accessed in the end of this seminar. So please join this webinar until the end to get the certificate. Let's start this event, which is our agenda today is welcoming speech that will be delivered by the head of Food Science Study Program, Department of Food Science and Technology, IPB University. I would like to invite Honorable Professor Dr. Harsi Dewantari Kusumaningrum. Please, time is yours.
Thank you, Nurafi. Thank you, MC. Assalamualaikum warahmatullahi wabarakatuh. Honorable Professor Hiroshi Kansaki from Okayama University, distinguished colleague, I see Dr. Nurheni, Dr. Endang Prantimurti, etc. Ladies and gentlemen, students and all participants of this webinar, good afternoon. Alhamdulillah, today we can meet together through this online media to listen an important webinar with the topic on polishing of a diamond in the rough, microbial bioconversion to create bioactive and natural products. It is a very interesting topic, I think. Indeed, it is an honor for all of us. This afternoon, we can welcome and listen our guest speakers, Professor Hiroshi Kansaki, from the Laboratory of Chemistry and Bioactive Compounds, Okayama University, Japan. Thank you, Prof. Hiroshi Kansaki, for being willing to share with us the current issues on the microbial bioconversion to create bioactive natural products. This webinar will give an insight and knowledge for academicians, particularly students and alumni of Food Science Study Program, IPB University, related to various aspects of microbial bioconversion to produce natural bioactive compounds and their current development. I hope collaboration on scientific events like this will continue in the future. I thank also to our colleagues, Mrs. Uswatun Hasanah, MSc, as moderator today. Also the Food Science Students Forum, Formasip IPB University, that organized this important webinar in collaboration with the Food Science Study Program, Department of Food Science and Technology, IPB University. Thanks also go to all webinar participants from all over Indonesia, places from Aceh to Papua, as well as all those who help making this activity a success. We have been already having good collaboration with a lot of Japanese universities, and this time with the Okayama University. Hopefully, activity like this will continue in the future and give significances for the development of sciences in Indonesia, especially in the field of food science. Without further ado, by saying Bismillahirrahmanirrahim, the webinar event with the theme Polishing of a Diamond in the Rough, Microbial Bioconversions to create bioactive unnatural natural products, hereby I declare to be open. Thank you for your attention. Billahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh. Thank you.
Thank you so much for the professor for the speech. And now we will start our main session of this international webinar. It will be handled by our moderator, Ibu Uswatun Hasanah, STP MSI. She is a lecturer of Department of Food Science and Technology, Faculty of Agricultural Technology, IPB University. Without further ado, I would like to invite Honorable Ibu Uswatun Hasanah, STP MSI. Please, time is yours.
Thank you very much, Ms. Nurafi. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillah, assalatu wassalamu ala rasulillah. Good afternoon, ladies and gentlemen, all the honored professors and our lecturers and all the participants. Thank you for your time to join today's webinar. My name is Uswa. I'm a junior lecturer in Food Science and Technology Department, IPB University. And now I'm also currently a student in Okayama University. It is an honor for me to meet all my lecturers here and also my friends here because I'm also a student, so I address all of you as my friends. So I'm very happy that today I would like to introduce all of you to Professor Hiroshi Kansaki, the speaker of our webinar today and also my supervisor for the 3 years here. So I would share a little about Professor Kansaki's curriculum vitae here. Professor Kansaki has awarded bachelor until doctoral degree in Kyoto University, and he was also a visiting scientist in National Institute of Health, USA, and since 2004, he has been a full-time professor in Okayama University. And for the professional experience, I would enlighten some of this. So he was a dean of Faculty of Agriculture, also the dean of the Graduate School of Environmental and Life Science, Okayama University, and the recent one, he was a former vice director of Okayama University, the vice president for international affairs. Professor Kansaki has an expertise in search and production of useful bioactive compounds using cells or enzymes. And I checked in ResearchGate today. His publication was over 100 publications. And here are some of the key scientific publications by Professor Kansaki. And he also collaborates with international scientists, such as from Thailand and also from Indonesia. So the topic today is very interesting. But before we learn together from Professor Kansaki, I would like to inform that after the presentation session, there would be a question and answer session. So please participate directly. So you, after this, can raise your hand and directly ask to Professor Kansaki. And Kansaki Sensei also told me that question in Bahasa Indonesia also okay. So I would try my best to get the point and transfer it to Professor Kansaki, and also from the chat room. But before going further, let us focus on the presentation first. So I would like to invite Professor Kansaki. Time is yours. Yoroshiku onegaishimasu.
Yep. Nice to meet you, all the participants. I'm very happy to join this international web seminar. I'm a first time to the international seminar. A little bit worried about a good presentation or not. Anyway, I'm very happy to have a chance and then this international seminars. Everybody can see my PowerPoint, okay?
Okay.
Yes, yes. Okay, clear.
Okay, just started. My title already, everybody said, polishing out the diamond in the lab. Microbial bioconversion to create bioactive unnatural natural product. This is a very rare word of unnatural natural product. Everybody knows a natural product. This one comes from the plant origin or the microbial origin. But in our case, bioconversion using enzymes or cells. This one, not naturally occurring product, but comes from the natural. That's why I mentioned the unnatural natural product. Okay. You can see that this is Okayama. You can see the Tokyo here and also there. Okay. Polishing diamond in lab, and everybody knows diamond is a very important, and everybody likes brilliant diamond. But cutting of a diamond is very important. The rough diamond needed diamond cutter to produce a brilliant diamond. Okay. Oops, sorry. Oh, something wrong. Okay. In Bahasa Indonesia, Berian Casa Porandia. This is correct? I'm not sure that I checked some dictionary.
Yes, Sensei. That's
Okay.
correct.
In today's presentation, I mentioned about a microbial enzyme catalyzed conversion of natural product to bioactive compound. A natural product in the sometimes a lab, and to bioactive compounds in the brilliant diamond. Okay. Firstly, our laboratory research outline. Search for high functional compounds and establishment of their effective conversion from low functional compounds. Okay. You know that many bioresources have a bioactive compounds. Natural product chemistry is a search for the bioactive compounds from bioresources, plants, and or microbial origin. But in our lab, okay, also search for the cell or enzyme. This cell or enzymes can convert for the low functional compound to the bioactive compounds. This research work is very useful for the biochemical probe or medicine and agrochemicals. Using these compounds, we try to mechanism elucidation of life phenomenon or utilization for sustainable agriculture or response for aging societies. This means power of microbes polish the rough compounds to produce useful products. I summarize today's topic. Four topics. First one in the anti-cancer drug, and the other one in the antioxidant compound, and the other one is a mushroom-treated woody biomass. And finally, I try to present our graduate school students' studies, including Uswa-san's studies. Okay, first one in the novel anti-cancer compound by actinomycete enzyme catalyzed conversion of fungal metabolite. you can see that this compound named albanolsin. This one is a very old compound, maybe 1960s. Some researcher already found this compound. We also found Streptococcus aureus isolated in Okayama universities. This one also can produce these compounds. I will show you that these compounds bioactivity. Maybe many researchers know the sea urchin. Sea urchin is a very famous meal in Japanese sushi or something like that. But in our lab, using a sea urchin test, you can see that this is a DNA in the female nucleus and the male nucleus. Just after the fertilization, female nucleus, male nucleus to near, and also the join, normal fusions, okay? But in presence of our compounds, after the 4 minutes, no fusion occurred. This means that no cell division occurred. This is a very easy to check the anti-cancer drug test. Some anti-cancer drug has activity in the cell division inhibitory activity. Okay. And also that we found that this albanolsin in the structure, 2 double bond here and there. You can see this one, cycloleucyl and phenylalanine. This is a 2 amino acid. This is a phenylalanine, and this is a leucines. I think, firstly, this albanolsin biosynthesized from CFL, cycloleucyl phenylalanine. But nobody knows that this kind of biosynthetic pathways. We found that this CFL oxidase, novel unique enzymes, catalyzed to dehydrogenation, this one and that one, and finally produce albanolsin. And also found these enzymes have a very wide substrate specificity. Many types of the amino acid residues also converted to the dehydro compounds. Firstly, we try to convert using this enzyme to produce some dehydro compounds. CFL is a phenylalanine leucine. This is a phenylalanine, glycine phenylalanine, alanine phenylalanine. These compounds we synthesize and try to bioconvert it. All of the compounds converted dehydro ones. This one is the both side dehydrogenation and one side one. Yellow one, we isolated the compounds and checked the activities. The summarize of the activity test. You can see the delta means the double bond. So that this one is the original compound, substrate. This is a one double bond insertion, the other side one double bond insertion. This one is the both double bond insertion. This slide mean you can understand easily. 2 double bond insertion has activity. This one albanolsin. And this one phenylalanine is also active and more potent activity compared to the albanolsin. Okay. This is a summarized one. Dehydro CDPs other than albanolsin can be synthesized by CFL oxidase. Tetradehydro CDPs, tetradehydro means the 2 double bond insertion, are active, while didehydro CDPs are not. This is 1 double bond CDPs. Activity of tetradehydro CDPs varies with their amino acid residues. Okay. Using this enzyme system, we think more active compounds could be prepared. I checked some reported papers. I found that some company research group found a phenylhistine. This one comes from the Aspergillus ustus, fungi metabolite. This one has phenylalanine and dehydrohistidine moiety and additional isoprenyl groups. Okay. This one double bond insertion cyclic dipeptide, but already they mentioned about the inhibitory activity cell cycle. This compound, abbreviation PRH. PRH is a didehydral CDPs, but active. This means if we can succeed to preparation of the dehydro PRH, could show the highest activity. I suppose that. We will try to bioconversion of this PRH to dehydro PRH, and also no isoprenyl group phenylalanine and histidine, CFH. We succeeded to preparation dehydrophenylhistidine and the C delta F delta H, and also the C delta FH. Okay. These compounds, what kind of the activity has? This is a very interesting point. Okay. This is a result for the sea urchin embryo test. Original compound phenylhistine in the activity. Very interestingly, one double bond insertion 1,000 times higher compared to the original compounds. Okay. And no isoprenyl group compound CFH, and also the one double bond compound C delta FH, no activity. This double bond 2 insertion C delta F delta H has activity, but lower than delta PRH. This means isoprenyl group and also the 2 double bond insertion needed for the potent activity for the inhibition of the sea urchin embryo divisions. Okay, and this is a result for the tubulin inhibition activities. You know the Taxol. Taxol in the plant-derived compound in the famous anti-cancer drug. And also the vincristine, also the plant-derived alkaloid. This one already commercialized anti-cancer drugs. These compounds in the 10 to the minus 7 molars activity has. You can see that this one, tubulin polymerization. Taxol inhibit the degradation of tubulins. Too much tubulins here. On the contrary, vincristine not produce a tubulin polymerization, different mode of actions. Our compounds, similar level of the 10 to the minus 7 or 10 to the minus 8 molars, potent activity, and similar to the vincristine-like compound activities. Okay, these results depend on these results. We get a patency to the PC international patency. Okay. We found that this delta PRH has a possibility for the anti-cancer drug. United States pharmaceutical company interested in these compounds, and they try to analogs. Now, plinabulin and the analog of the delta PRH is succeeded to the test for the anti-cancer drugs. Now in the clinical trials, phase 3, by the Beyond Spring companies. last years, they said that now in the positive top-line result from its protective 2 phase 3 registrational trial of plinabulin in combination with pegfilgrastim for the prevention of chemotherapy-induced neutropenia. This is a homepage in the companies. The compound plinabulin and some first-class stem cell modulator binds in the vicinity of the cohesin binding domain of beta tubulins, and also the immune defense protein, something like that. If you're interested in, please check the homepage. I am a microbiologist. I am very interested in the microbial conversion. This is one of the examples. Many people has hyperlipidemias. That's why the anti-hyperlipidemia needed. One of the company, Sankyo, commercialized first statin analogs, pravastatin sodium. This one originally comes from the product of the fungi, compactin. This one active, but something problem for the commercialization. But Sankyo company succeeded microbial conversion, and this is hydroxylation and opening some ester bond. Finally, they succeeded to the production. The compound inhibited HMG-CoA reductase and lowering cholesterol levels. This is a combination of the two microbes. This is our result, our lab's result. This is a fermentation product by fungi and microbial conversion by another microbial enzymes. And the Sankyo's data also the very similar results. This is a two-step fermentation. That's why this is a natural product. This one is an unnatural natural product. Okay, next topic. First topic in the microbial product conversion by microbial. Next one in the plant secondary metabolite converted by microbial enzymes. Okay, everybody knows that olive. Olive is a very famous one for olive oils, especially in the European countries. But recently, many Asian countries using Italian food or the other kind of the foods. But only olive fruits for eating. But leaf is limited use for the some things. Only the some country use for cosmetics and supplement. Olive leaf has a famous secondary metabolite named oleuropenes. This one is attached to the glucose. And this one metabolite in one of the metabolite hydroxytyrosol. This one used in a whitening agent in some companies. Okay, but we try to convert these compounds to the another more high functionalized compounds. We found baker's yeast, very common fermentation microbes, converted one of the compound in the olive leaf extract to produce a compound B. We isolated both compounds, and finally we found that baker's yeast catalyzed the compound A to compound B's reactions. And we isolated both compounds and determined to the structures. This is a compound A structures and this is a compound B structures. You can see only the here and there changed by baker's yeast. This is the aldehyde group. This is the alcohol group. This is a reduction. Reduction catalyzed by baker's yeast. Other part no reaction occurred. Bioconversion is a very useful for the only the some part of the compound change. This compound and the original one in the glycoside deglycosylation in the this aglycone produced and non-enzymatically change this compound. Okay, and this compound has some special phenolic OH. This is an important part for the antioxidative activities. We checked the many types of the antioxidant activity. ORAC is a very famous for the United States governmental decision of the antioxidant activity, and DPPH, very old type of the antioxidative activity. You can see that this is a AANA, mean the ascorbic acid sodium. This is a very famous antioxidant activity compounds, and also the Torox, also the vitamin E. These two compounds is a very much known everybody, but this compound, some of the tests, not so much activities. Compound B and A has a very potent activity compared to the ascorbic acid or the Torox. That's why that this is a meaningful for the commercial utilization of the compound B. Next one. We use the baker's yeast for the conversion of the compound A to the compound B, but some problem. Conversion rate is low when converting with baker's yeast. That's why we try to search flower-derived microorganisms with higher conversion rate. Because, as you know, many flowers has the origin of the yeast. Yeast can convert maybe this reduction reaction. 30 kinds of flowers, including 5 variety olive flowers here, I selected and also try to isolate the microorganisms. Okay, including peach, plum, or something like that. And this is a method for the enrichment cultivation to isolate flower-derived microorganisms. We succeeded to isolate 196 strains and check the bioconversion abilities. We found 3 isolate from olive flowers has the high activity strains. All of the 3 ones identified to be black yeast, Oleobasidium plurans. These 3 strains has activity, highly conversion, and we identified these bioconversion one the same as compound B. This is a result for the conversion. Produce amount here, black circle and isolate from olive flowers. And NBRC strains are same Oleobasidium strains. And Saccharomyces cerevisiae is same as a baker's yeast. You can see that these 3 strains much higher activity compared to the baker's yeast. In the cosmetic preparation, bread smells is not so good, but olive leaves derived flowers strains, no smells. And also the image for the olive flower derived strains catalyze olive leaves extract to produce a new cosmetics. That's why one of the company in Okayama is developing cosmetics containing these compounds. Okay, next one. You know, in the world, maybe many researchers focused on the environmental things. Woody biomass used for the environmental more free things. That's why our laboratory also try to woody biomass utilizations. In Indonesia, also the timber production, many timber productions, but sawdust and the woody chips, low utility values, only the burning or some other things. As you know, in Japan, many milk cow, dairy cow, breeded, but their breeding, all of them are imported. That's why Japanese government try to use the domestic feedings. As you know, that TMR means total mixed ratios. And the animal researchers focused on the lactic acid fermentation. This is the fermented mixed ratios. Okay. Bagasse is a famous additives to the TMR, fermented TMR. But bagasse also imported in the case of the Japan. But in Indonesia, maybe many bagasse. That's why that instead of bagasse, we try to use the woody biomass. Okay. But some problem using woody biomass containing a cellulose or the hemicellulose. These compounds degraded by cow or the cow microbials one. But the lignin and the tightly bound to the cellulose and the hemicellulose. That's why the cow cannot degrade it directly or the woody biomass. Okay. How to solute? Degrading lignin by white rot fungi. This is the mushrooms. Edible mushroom can degrade the lignin, and remaining cellulose or hemicellulose may be easy to degrade by cow. Okay. And also, this is a cascade use. This is the Okayama prefectures, and Usua-san and I live here in the Okayama cities. Okayama prefecture, northern part of the Okayama prefectures, is a tree farm. Maybe many trees here, timber production here, and sawdust. Using a sawdust, edible mushroom production, and mushroom bed in the fermented TMR production. And near the tree farm, many livestock areas. These cycles near the land. Maybe from some tree farm to the another cities, it needed a more costful. That's why this cycle is very important. This is our research result. Hinoki is a cypress. This is one of the famous trees in Okayama or in Japan. Hinoki itself, here, 100% hinoki, almost cannot degrade the cow, 1.2% biodegradability. But after shiitake treated, this kind of thing, more than 15% can degraded by the cow-derived enzymes. This is a similar level to the bagasse. That's why shiitake treated woody biomass can be used for the TMR productions. Okay. And I asked TMR, fermented TMR preparation and feeding test. You can see that this is a shiitake mushroom production. This one is a mushroom bed containing many types of the woody biomass. After the cultivation, this is a remaining mushroom bed. We try to produce this TMR. We use a 2-ton woody biomass degraded by edible mushroom was used, and a 30 kilogram, this one, too big one. And feeding 40 dairy cows and automatic milk making by robots. This is a lactation performance. This is a no addition of the shiitake mushroom bed, milkweed, butterfat, milk production. Second week, 2% containing, woody biomass containing TMR used feeding. Not so much different in some performance. And 5% addition of the TMR, also not so much big difference. This means the softwood sawdust degraded by edible mushrooms would be available as ingredient of fermented TMR. Okay. This is a first part of the woody biomass utilization. But this one also not so much expensive one. We think more better utilization. In Japan, many people has a pet like a dog and a cat. Especially recently, many small dogs available in families. Many people used much money for the pets. Drinking water, very expensive feeding or something like that. We know the mushroom culture ones, sometimes bioactive compounds. We try to check enoki mushroom bed before the cultivation, after the cultivation. Anti-obesity. This one highly active. This means after the cultivation, enoki mushroom converted some compound to the highly active compounds, or the enoki mushroom itself has some active compounds. Anti-obesity in the humans problem, not only the humans problem, but also the many pet problem recently. That's why after checking the activity, we produce some commercialized pet drink ones. Okay, this is a result summary. This is the inhibition percent of the fat anti-fat producing activity using some special animal cells. Okay, blue one in the untreated cypress, red one in the enoki cultivated cypress. You can see the much increasing activity. This is one of the example for the lipid dyeing test. Red color in the lipid accumulation. 25 microgram per ml treated one, no lipid accumulation. 5 microgram per ml one in the very small amounts of lipid accumulation. We decided the 5%, 50% inhibition concentration, IC50, untreated cypress also has activity. 0.1 milligram per ml. But you can see after the enoki mushroom cultivation, more than like 20 times higher activity. Okay, this means enoki mushroom converted some compound to the active compound, or the enoki mushroom itself produce bioactive compounds. We check the activity animals itself. We use some special mouse and drinking test. Test group 1 in only the water. Testing 2 group in the water plus preservative. And 3 the water plus preservative and mushroom extract. Okay. After the 12 weeks drinking test, you can see fat cells shrink here. Only the water fat accumulated. Okay. And also the interestingly, analysis of intestinal flora, only the water drinking test group, intestinal bacterial diversity is too small. Plus extract, higher diversities. Recent research result, maintaining the diversity of intestinal flora is essential for healthy life. This means the extract has some activity in the maintain the intestinal flora diversity. These results assisted us to produce a commercial pet drink. Now, some company want to try to this kind of the commercial product. Okay. From now, I will summarize our group research topics. From now on, I inform the PhD students in our labs the result I will show you. Dr. Panarat Arunatiakon, she comes from the Thailand, Sinakarin Wirot University. Now she's a teaching staff in the Sinakarin Wirot University. She got a PhD degree in our lab. After returning back to the university, she focused on the bioconversion. And Thailand, also Indonesia, has many types of the tropical fruits. Famous one is the mangosteens. Many Japanese like mangosteen fruits. This is the queen of fruits. But outside of the mangosteen has some special compounds. Alpha mangosteen, the compound names. She tried to bioconversion experiments. Added to the fungi cultivation of this compound, and isolation of some bioconverted product, and structure activity relationship, and check the metabolism studies. She focused on the endophytic fungi isolation. She also used our lab's Orea europea, olive leaves. And I don't know, not so well, but this one is a very famous plant in the ASEAN countries, Thylacorana triandra. She used these two plants and isolation of some special fungi. Endophytic fungi, many researchers focused on because this kind of fungi can convert the plant secondary metabolite to some other compound. That's why she thinks there's some possibility these microbes has the activity convert to some special compounds. Okay. One of the result. This plant-derived isolate to isolate different bioconversion. But you can see that this is a sulfonation and also that this is a hydroxylation. Two types of the derivatization she found it, and all of them she isolated. Different fungi has a different conversion activity. Okay. And also another bioconversion, olive-derived species. Compared to the previous slides, this strain has a different bioconversions. This side of the hydroxylation, and this one is hydroxylation, and this one is the cyclizations. Okay. And she checked biological activity. This is one of the example, and the antimicrobacterial activity. Original substrate as a MIC in 6.25 micrograms. These three compounds inactive. But interestingly, these compounds after the sulfonation 2 times higher activity. This is a metabolism is very interesting, but some possibility has a more potent activities one. Both aspect, more active compounds preparation and also the metabolism research interesting for the scientifically. Okay, finally, I will present Usura-san's presentations. She focused on the mangiferarin compounds. This one derived from the mangoes. You can see in the glucose attached to the some A-glycon, but then CC bond. C-glycoside is difficult to cleave. Only the 2 microbes, intestinal bacteria, converted mangiferarin to nonnatriol. This one, different bioactivity compared to the mangiferarin, and sometimes higher potency. That's why we want to prepare the nonnatriol more easily. For that purpose, we have to isolate some active strains which catalyze these conversion reactions. She succeeded active strain from the mouse feces. Several types, 4 different types of the strains isolated, but this is a same genus and same strain and same species strains. Okay. And after the identification using a 16S RDNase ones, we decided these strains belong to the Bacillus strains. A 99.2% similarity to the Bacillus hisashi and 99.0% similarity to the Bacillus thermoamyloborans. But no strains completely same. Interestingly, both strains is a facultative anaerobic and thermophilic, but an already reported mangiferarin's conversion strains and the obligatory anaerobic and also the not thermophilic strains. That's why that she tried to check this strain can grow under the aerobic conditions, also undergrow more than 50 degrees Celsius or something like that. She checked various cultivation methods anaerobic condition, aerobic condition growing, and shaking static, and the reaction condition also the anaerobic or aerobic. Interestingly, the strain can grow aerobic better than the anaerobic. And more interestingly, aerobic conditions growing cells has activity. And also aerobic conversion, also we can check it. This is the first report of mangifferin deglycosylating bacterium that grew and converted under aerobic conditions. And also more than 55 degrees Celsius can grow and also has activity. 50 degrees Celsius optimum growth, 55 and 60 good growth and activity remaining. All cell grown at 50 degrees showed high level of unnatural in 24 hours. And also interesting, similar genetic strains we obtained and check the growing. Bacillus hisashi, Bacillus amyloborans grow at a 37 degrees Celsius and 50 degrees Celsius and aerobic condition. But all of the three strains has no activity conversion mangifferin to naratirol. That's why our strain only can convert it aerobically mangifferin to naratirol. In future, using these cells, we try to produce some naratirol. This experimental work also collaborated with some Japanese company. That's why the Uswa-san and I have some inventors for this one, and we already applied the patent. This is a summary of the uniqueness. Bacillus species KM1 very similar to the Bacillus hisashi and Bacillus amyloborans, but these two strains no conversion. Our Bacillus species KM1 in the conversion. Previously reported mangifferin converting bacteria only growth obligate anaerobic and anaerobic conversion. That's why our strain is a very much uniqueness. Okay. And finally, this is some homepage from the Umami Information Centers. Everybody knows Umami. Umami is a Japanese, but nowadays English also Umami. I can share the YouTube's video, maybe 1 minute or something. Oops. Oh, sorry.
Sorry, Sensei, maybe need to play again.
Okay.
Only a little sound and then stop. Thank you, Sensei.
That's okay.
We still cannot hear the voice yet, Sensei.
Really? We will try again. Okay.
Thank you, Sensei.
You can hear it?
Okay, Sensei.
Okay.
Basics.
Sorry, Sensei. Now the sound is very low, so we can
Very
hear
low?
but only a little. Yes, Sensei.
I'm not sure.
Or maybe if because of the connection, it's okay, Sensei.
To comprehensively determine the
now
taste
okay,
of
Sensei,
food.
clear.
How about it? Okay?
So sometimes we can hear, but
One of the important factors that
now okay.
determine the taste of food. Amino
It's better?
acids support the lives
Sensei.
of all living organisms on Earth. And all creatures need amino acids in order to live. Among the many amino acids that exist in the natural world, it is glutamate that is the most abundant. That's why it is used in a wide variety of foods. And the taste of this glutamate is what is known as umami, one of the basic tastes, along with sweet, sour, salty, and bitter. Glutamate is included in many familiar foods. Through these foods, we come in contact with the taste of glutamate, or umami, on a daily basis. And umami, produced by glutamate, has a characteristic of becoming 7 to 8 times stronger through substances such as inosinate and guanylate. Inosinate is abundant in meat and fish, and guanylate is rich in dried mushrooms. Let's take a look at the combination of tomatoes and meat. As with cheese and dried mushrooms, foods taste better when combined with other foods. This is the synergetic effect of umami. So now you're asking, what kind of taste is umami? Let's look at 3 of its features. 1. It spreads all over the tongue. 2. It lasts longer. 3. It facilitates the secretion of saliva. Let's try umami. Umami history. In 1908, Dr. Kikunae Ikeda noticed that there was another taste other than sweet, sour, salty, and bitter. While studying abroad in Germany, he found a common new taste in tomatoes, cheese, asparagus, meat, and other foods. After returning home, Dr. Ikeda succeeded in extracting this new taste from Japanese kelp soup stock. He discovered that it was composed of glutamate and named it umami. For centuries before umami was discovered, many fermented foods and traditional seasonings were created throughout the history of the world, and they were devised for making food more delicious or preserving food. In addition, the drying and salting of food was devised to concentrate the umami, and fermentation was also a means to increase the umami. In ancient Roman times, one of the 3 major traded goods actively produced along with wine and olive oil was garum. Garum is a fish sauce made by fermenting small, salted fish. Factories and shops of garum once existed around Europe, mainly on the Mediterranean coast. These are said to be traces of the earliest food industry in the history of mankind. Fish bones and glutamate have been detected in the soil of amphora pots used to store garum, which were excavated from the Pompeii ruins. Since then, umami spread into fermented beans, grains, seafood, cheese, and tomato products, and it is today included in everyday food for people everywhere. Furthermore, the health value of umami has recently been attracting attention around the world. Yesterday, today, and tomorrow, umami, including glutamate, continues to create tastiness around the globe.
油脂を中心に他の国は料理を構成してますけども、唯一日本だけがうまみ成分を中心に料理を構成したと。それによって、解析で言いますと、解析は、63品目あって、1000キロカロリー。フランス料理ですと23品目で2500キロカロリーというように、非常にこう、カロリーに差が出てくる。そのことは、それから、人類の、健康に非常に貢献するであろうというように、思ってますけども。日本料理を広げるということは、その旨味の文化を広げるということイコールですから、昆布とカツオがないと日本料理が作れないというのではなくて、まあドライトマトと、ドライモリー油で精進の出汁もできますし、チキンの胸肉と、ドライトマトでも、グルタミン酸とイノシン酸で相乗は起きますから、そういう意味では、いろんな食材で、そこの国にある食材で出汁は引けるというように考えてます。うまみとは、世界の先進国の肥満を、救う、非常に、重要な、ツールであるというように考えています。
So you're lucky you discover umami today. Me, I wait until I'm 40 to discover what it was umami, okay, 2 years ago. So if you think you will find a lot, okay, in my original cooking is not Japanese, it's French. So after the first course I get on umami, I think all what I did before, in chicken stock, veal stock, demi-glace, there is umami. So, and now I'm using it the way to first reduce the salt on the dish and to balance the dish. If I compare the dish I make 5 years before and now to use these techniques, the dishes are more balanced. There is more balance on the flavor, and I think this is the way I'm using the umami flavor, is to balance the dish. There is more balance on the dish we create now. And this is what, with Chef Nobu, started to do some umami dinner, to explain to the guests what is umami. And all our new recipe now we create for Nobu around the world is all based on umami.
Sono Gabriella Morini, sono un ricercatore in chimica organica e qui ci troviamo all'Università di Scienze Gastronomiche di Pollenzo in Italia. In questa università io insegno un corso sulle basi molecolari del gusto, cioè su come funziona l'apparato gustativo. Tra i gusti quello che è meno conosciuto e che in Italia non viene mai considerato è l'umami. Questo perché viene ritenuto che sia un gusto tipico della cucina asiatica e non di quella italiana, e quindi tra gli altri mi occupo anche di spiegare cos'è l'umami. Perché è importante studiare l'umami e spiegarlo? Perché l'umami è una delle principali caratteristiche che rende gradevole un alimento. Infatti i sinonimi sono savory in inglese, saporito in italiano. L'umami è di fatto il gusto più umano.
Umami Basic.
As you know, the umami in one of the famous Japanese word, and also the umami in the food science in the very important ones. In the previous video mentioned about umami is discovered in some special professor in Tokyo University. This is one of the result of the natural product chemistry. He found what's kind of the umami taste or something like that. And he finally found monosodium salt of the glutamic acid is the constituent of the umami taste. He isolated from this monosodium glutamate from the seaweed kombu. Everybody knows recently in the world, MSG produced by the microbes. These microbes also found in Japan, but worldwide production, of course, in Indonesia, maybe some company produce MSGs. And amino acid, some amino acids are very important for our food and also the anything for the cosmetics or other things. This one produced by the microbes. That's why the microbial fermentation is very important for our lives. Okay, and our laboratory results, also the microbial powers converted some functional compound to the more highly functionalized one. Okay, maybe in Indonesia or the ASEAN country, also Japan has a much microbial resources. That's why there's some possibility to produce more functional food or other things. Usa-san, continue to some other videos. That's okay. Enough time to the continuance.
Maybe still enough, Sensei.
Okay. And also another video in the ambitions. This one in the Professor Ikeda Kikuna's drum. This is not scientifically ones, but this is a very impact to the students or the young lecturers. That's why I selected this one.
Excuse me, Sensei, for before playing the video, is there any option for also share video, Sensei? So not only share sound?
Okay. Share the sound. This one can hear?
Yes, Sensei, so we can hear
This is the story of a
voice. Yes, Sensei. So
okay,
okay.
in the large scale of the video can share or not?
Yes, Sensei. Would you please try to
this one okay?
play the video?
Sound is low?
So now we have not hear the sound yet, Sensei.
This is the story
This
of a
one is
man
better?
who made a great
So
discovery
the
almost
sound
exactly
is
100
okay, Sensei,
years
but the
ago,
video is
when
not
Japan
yet
was
played in
undergoing
our
tremendous change.
Okay.
view here.
If so, we not continue to the video. Okay, another story.
Okay,
one.
Sensei.
Yep. Okay. Finally, I presented in a power of microbes as a tool of the polish and natural products. Maybe I only checked the Indonesian foods in the fermented one, and this is one of the example I checked in the Indonesian homepage. In the tempeh is a very famous fermented food. In this one, in the Rhizopus species produce one, and dadia and the fermented milk, and pakasam in the fermented fresh waters, brem in the fermented glutinous rice. This one is a baker's yeast, Saccharomyces cerevisiae yeast. Onchom and maybe I don't know exactly, some special plant. And this one, danke, the fermented buffalo milk, and tape, the fermented cassava. Maybe Uswa-san focused on research work in this one. And also the terasi is a fermentation in Indonesia, shrimp paste. Maybe other types of Indonesian fermented foods. This is some treasures of the microbes. Some researchers isolated these special microbes and try to use this one. But I think, sorry. Fermented foods are known to be indispensable to Asian foods containing a variety of microbes. We have much possibility to effectively use treasure buried in nature by fermented food microbes. That's why using isolated one, try to convert some functional compound to the much more higher functionality or something like that. And also the fermentation, new fermentation product, also some possibly produced. Okay, that's why I hope the many Indonesian researchers focused on the microbe itself and also the microbial conversion using Indonesian domestic plant or the fish or the other things, and check the functionality to good for the human health or sometimes the pet health or something like that. After the Usua-san and we return back to the Indonesia, I hope that we will continue to collaborate with the Indonesian researchers and the Japanese researchers. In our group, Professor Nitoda is a specialist for the structure determination of the natural product. And my major is applied microbiology using enzymes and using microbes. I hope to continue to the collaboration. And finally, this is a photo of the 2018 and the 2020s, our laboratory's graduation ceremonies, the students. Here is one of the Thailand students. He got a PhD and he worked in a Thailand university. And Usa-san here, and Usa-san's husband, Sai-san here. Both of them got a PhD in our university. They will soon return back to the Indonesia. And other master course students and also the undergraduate students continue to do the research work. Maybe these students, full of love, they continue to do the research work and the company wants, and maybe be a diamond. I think today's lectures, many students in Indonesia, IPB, and also the young lecturer or attended the meeting, they also sometimes laugh. But some professor already brilliant diamond. But I hope all of the students in Japan and Indonesia will be a brilliant diamond. Terima kasih atas perhatian Anda. Terima kasih. I have enough time to discuss about my lectures, and also if you need some more information about YouTube or something like that, I will send an email or something like that. That's all of my presentation. Thank you so much. Terima kasih.
Thank you.
Thank you very much, Professor Kansaki, for the very interesting explanation. So we already broaden our knowledge about the utilization of microbes, also related to food, to increase the human's quality further. So next, we would like to start the question and answer session. So here, there are some questions in the chat room, but I would like to invite to directly ask. So here, Mr. Azmi Azhari from Pajajaran University, would you like to ask directly to Professor Kansaki, please? For Mr. Azmi Azhari. Okay, or is there any other participants that would like to directly ask to Professor Kansaki? Okay, so while waiting, I would like
something.
to... okay, please, Professor Hanifah.
Thank you.
Yeah.
Nice to meet you, Prof.
Nice to meet you. Yeah.
I like to know the chemical compounds you identify uh, after bioconversion. I like to know the way to identify the molecules after bioconversion. You identify first the molecule, or you check the activity first, then identify the molecule, and how to isolate. Uh, is it the isolation also uh, like activity guided fractionation and isolation?
Okay.
Is it the way like that or any other way? Thank you very much.
Yeah. Thank you for your questions. First thing in first isolation or the first activity check, and it depend on the experimental work. In the case of the first topic on the anti-cancer drug, I use a isolated enzymes, purified enzyme, and also this one in the have the activity to the many types of the compound. That's why in the case of the natural product, we isolated first and isolated compound conversion using a purified enzyme. This is a very simple system. If so, the very easy to purify. That's why after the purification and identification and checking the activity. But using extract for the plant-derived extract, like olive leaf extract, in this case, we are not sure what kind of the compounds will be the substrate or not. That's why the mixture of the extract treated many types of the microbes. After the conversion, we check some
Activity.
conversion occurred or the activity check. The both checked using a mixtures. If the activity unique or some chromatography, some checking some new compounds occurred. If so, the both directions, purification of the new compounds and also the activity guided fractionation.
Okay.
Okay. After the purification, we use many types of the analytical method to determine to be a structure and also the more detailed information for the biological activity. You understand? Okay.
Yes, I understand. Thank you very much.
Yep.
Actually, I like to know one of your explanation about how to check the different activity from the olive
Oh, yes.
extract.
You mean
The
the
different activity. Yeah.
antioxidant activities? You mean different activities?
Yes. You check many microbes, right? With the olive extract. How
Yes.
to check the bioconversion result? Is it by FTIR spectra or by UVV spectra or roughly?
In the case of
I
the screening, we use some TLC chromatography first, chemical checking. First, the compound B to compound A to the compound B conversion. If so, that we know the HPLC profile or the TLC profile.
Okay.
But after the
Just
cells
profile.
conversion, check what amounts conversion occurred and a detailed information checking or something like that. You
Okay.
understand?
Just the profile only with maybe some absorbance.
Oh, yes. In our lab, we have some special type of HPLC and detected multi-wavelength detectors. That's why we check a UV spectrum, separation and a UV spectrum understand. If so, the same UV spectrum, the same some analog or something like that. Easy to understand or something like that.
Okay, thank you
Yep.
very much. It's very clear. Yes.
Yep.
If the profile is the same, so there is no bioconversion. But if the profile is different, especially from SPLC-DID, right?
Yep. Yes.
So the bioconversion occurred. Okay, thank you
Yes.
very much, Prof.
Thank you.
Sensei.
Yep.
Very clear. Thank you.
Thank you so much.
Yeah.
Thank you very much, Professor Hanifah, to also make us more clear about the technical details for this experimental work. So next, I would like to invite Mr. Ahmad Wildan Muhammad Putra. So please say your question.
Thank you, Ibu Uswatun Hasanah, for giving me chance to give the question to Sensei. Hi, Sensei. Thank you. I have two questions, Sensei.
Yep. Yep.
One question is about the olive oil. As you mentioned, olive on your slide presentations, olive oil has activity as antioxidants and antimicrobial as well. So what do you think? Which one the most potential of olive oil, whether as antimicrobial and antioxidants? That is the first question, Sensei. And the second
First question, please. Once again, please.
Okay.
Yep.
As you mentioned on your slide presentations, if olive oil has activity as antioxidants and also antimicrobial. In Indonesia, comparing to other oils, olive oil is quite expensive. So what do you think? The question is, which one the most potential of olive oil, whether olive oil as antioxidants or antimicrobial? That is the first question, Sensei, and I have
I understand. I answered the first question. That's okay.
Okay, Sensei.
Your question is olive oils ones, but I use not olive oil, olive leaves. In the plant, some soybean, also the soybean oil and the soybean itself. Oil has a fatty acid, lipid. This one dissolve in the secondary metabolite. Secondary metabolite, sometimes hydrophobic compounds, not soluble to waters. That's why the oil containing a hydrophobic secondary metabolite. In the case of the olive produce oleuropein or the other antioxidative secondary metabolite. That's why the olive oil also containing a special compound secondary metabolite. As you know, plant itself produce a secondary metabolite, but depend on the species. Olive can produce oleuropein. Other similar plants also produce oleuropein, but other soybean or the other oil plant cannot produce oleuropein. That's why sometimes depend on the kind of the oils, different biological activity. In the Mediterranean areas, much used olive oils. Maybe this one sometimes healthy or something like that. This one comes from the oleuropein or something like that. Many researchers focused on this one. But our result and also focus on the olive leaves. Olive leaf has a much more potency compared to the olive oils. That's why some other possibility we focused on. You understand?
Yes, Sensei, I got it. So can I deliver the question number 2, Sensei?
Yep.
Okay, Sensei, thank you. As you show us the video, even though they want only the sounds about umami.
Yeah.
Can I deliver the questions about umami, Sensei?
Yes, okay.
Yeah, as I will conduct a research about umami from one of the fermented food in Indonesia. But Sensei, I quite worry as one of the stage using panelists. To to test which one the fraction that has the strongest. Due to the COVID-19 pandemic, it's quite difficult to collect the candidates as trained panelists. So what do you think? Do you have any idea which can replace the sensory analysis in in conducting a research of umami? That's
I
thank
have
you.
not I have not a specialist for the umami checking the taste, but one of my friend in some checking some taste check, and sometimes computerized or some special machines already available, not panelist checking, but sometimes human power is also the information also important, but I'm not sure if I have a time. Please, I ask some my colleagues to solve these questions. Is that okay?
Okay, Sensei. Thank you so much.
Okay. Thank you very much, Mr. Ahmad Wildan, for the question. We still have time, so I would like to read the question from the chat box. So first, from Mr. Azmi Azhari, how to choose specific microbes for targeted activity such as anti-hyperlipid activity?
This is in our lab's result, but in the case of the screening of the microbes, firstly, selection is important. That's why the how-to activity focused on, and also the how many microbes we check is very important. In our case, in my student days, also I try to screen some enzymatic activity, more than 1,000 different types of the microbes cultivated and check the activity. This case, microbes derive the soil and plant isolation, and also some deposit microbes already identified one. That's why the firstly choose some activity, biological activity or the enzymatic activity, and also the screening of the many types of the microbes. For example, bacteria or the fungi or the actinomycetes and lactic acid bacteria. Many types of the isolate, try is important, I think. But sometimes important thing, some logical screening is sometimes important, but in my case, sometimes non-logical screening is some good results. In the case of the first project, and not logical screening, and many students try to screen the active compounds producer or something like that. That's why in the lab course, I'm not sure in Indonesia's case, Usa-san knows our third year students have a lab course and around 30 students participant. In my young days, my lab course in the isolation of some microbes. That's why young students sometimes unique microbes isolation. After the isolation, we try to cultivate to check the activity. Sometimes good results obtained. You understand?
Thank you very much, Sensei. So first is what kind of activity that we would like to target, and after that, the screening also depend on the microbe or the sources that we have. And the other question, is there any approach of targeting silent gene for microbial enzymes production?
I know that this one, but I did not try this one because I have some molecular biology technique, but I think important things, active microbial enzymes important, and still many active enzymes not screened in the natures. That's why the cultivated and active enzyme screening in my life. You understand? Maybe some molecular biologists try to check the silent genes approach.
Thank you, Sensei. And then the next question from Mr. Henry Noor. Thank you for the nice presentation, Professor Hiroshi. My questions are related to the mangiferin bioconversion. Actually, which enzyme involved in the bioconversion, extracellular enzyme or intracellular enzyme, and where the bioconversion occurred, inside or outside the cells, and which one is better to improve the mangiferin bioavailability by enzyme addition or by using fermentation?
Okay, this question is better to answer the Uswatan, but then I will do the answer the question. In our case, we use resting cell reactions because this kind of the compound degraded inside the cell's enzymes. You know, the alpha amylase or the cellulase or beta glucosidase produced as an extracellular enzyme in the bacterial case. But for the screening, extracellular enzymes are a little bit difficult to do that because an extracellular medium has a much another compound. That's why in our case, only use the resting cells for the enzyme sources. That's why I think mangiferin degradation also inside the cells or the near the membranes regions. Our result, Uswa-san's results, also very interesting in the some possibility in the membrane enzyme or something like that, but we are not sure. And I'm not sure, also not sure, extracellular enzymes or the intracellular enzymes, which are better to improve. But I think intracellular enzymes, some more possibility to improve. That's all.
Okay, thank you very much for the participants that already give the questions. Actually, we still have several questions in the chat room here, but due to the limitation of the time, I would close the question and answer session. So we already get the new points of view about polishing a diamond in the rough. So related to how we can utilize microbes for creating better compounds that after that we can apply in our daily life. So as a moderator here, I think my duty already finished, and I would like to return back to the MC, Ms. Nurafi. Time is yours. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much to Honorable Professor Hiroshi Kansaki and Honorable Ibu Uswatun Hasanah STPMSI for the very enlightening session. Please give applause.
Thank you.
And now it's time to announce the door prize for the best questions from two of the participants today. The selection is done by the committee based on the best of two questions during the webinar session discussion. Well, the first door prize is given to Asmi Azhari from Pajajaran University, and the second door prize is given to Henry Nur F. So congratulations for both of you. The committee will contact you later. And now, ladies and gentlemen, it's time for a photo session. So to all participants, make sure to turn on your cameras because there are some slides here. So just ready. I will count on 3, 2, 1, go. Okay, for the second slide, on 3, 2, 1. And then the next slide, on 3, 2, 1. For the fourth slide, 3, 2, 1. The fifth slide. 3, 2, 1. And then for the last slide. Ready? 3, 2, 1. Okay, thank you for your time and your smile. And now it's time to certificate handover session. The first certificate given to our moderator today, Honorable Ibu Uswatun Hasanah, STP MSI. So this is the certificate. Thank you very much, Ibu Uswatun Hasanah.
Thank you very much. It's my pleasure.
And then the second certificate is given to our main speaker today, Honorable Professor Hiroshi Kansaki. Thank you very much, Professor.
Thank you.
Okay, this is the certificate for you.
Thank you so much.
Well, ladies and gentlemen, don't forget to fill the absent in this link in chat room. Okay, now we come to the end of this international webinar. Thank you to our speaker and also to all of participants for joining us today. I am Nurafi Rasna Suhaima, representing all of the committee, would like to give you our deepest appreciation for your support in this event. And we do apologize for our mistakes in presenting this event. Thank you and have a great rest of your day. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank
Thank
you,
you very
Professor.
much.
Thank you.
Thank you very much, Sensei.
Thank you.
Arigato gozaimasu.